Hello, future doctors. Today we explore a fascinating topic. Monoclonal antibodies, or mAbs, are a big deal in modern medicine. These therapies offer new hope to many, as doctors around the world now use them. Think of these antibodies as incredibly smart keys. Each key fits only one specific lock, and in this case, diseases are like the locks. Monoclonal antibodies are these special, unique keys. They will only interact with a single target. This precise targeting ability is extremely powerful, helping to minimize harm to healthy cells. The development of mAbs is a story of brilliant innovation. Scientists dedicated many years to this research, aiming to create better, more effective treatments. So, prepare yourselves to learn a great deal. Your body has an amazing defense system. It naturally makes antibodies to fight invaders, which can be bacteria or viruses. The antibodies your body usually makes are polyclonal, meaning many different types work together. The term monoclonal tells us something crucial. It means all the antibodies are of one single type, originating from a single parent immune cell. Hybridoma technology is the secret behind making mAbs. Two brilliant scientists, George Kohler and Cesar Milstein, received the Nobel Prize for this landmark discovery back in 1975. They devised a very clever and effective method, allowing for the production of these special antibodies in large quantities for therapy. The production of monoclonal antibodies is a meticulous process. It begins with identifying a specific antigen, the target molecule for the antibody. Often a mouse or another suitable animal is immunized by injecting it with this chosen antigen. The animal's immune system then makes antibodies against it. Next, the antibody-producing B cells are carefully isolated, typically harvested from the animal's spleen. These specialized B cells are then ready for fusion, mixed with special laboratory-grown myeloma cells. The fusion creates new hybrid cells called hybridomas. A special chemical mixture called HAT medium helps select, allowing only the successfully fused hybridoma cells to survive. Finally, the stage is set for large-scale production. Section 4, Clinical Champions. Monoclonal antibodies transforming patient care. Cancer treatment has been profoundly transformed by monoclonal antibodies. Some monoclonal antibodies work by flagging cancer cells, making the cancer cells more visible. The patient's own immune system can then find and destroy them. Other monoclonal antibodies function by blocking important growth signals. Trastuzumab, also known as Herceptin, for certain breast cancers is a prime example. Rituximab, or Rituxan, effectively treats some types of lymphoma. Autoimmune diseases are also seeing remarkable benefits. Adalimumab, known as Humira, is widely used to treat rheumatoid arthritis. It specifically targets a molecule called TNF-alpha, which is a key driver of inflammation in arthritis. Infectious diseases represent another important area for monoclonal antibodies. Palivizumab, known as Synagis, is used to prevent severe RSV infection, particularly for high-risk infants. More recently, during the COVID-19 pandemic, several new monoclonal antibodies were developed. The applications of monoclonal antibodies do not stop there. Many other uses are currently emerging and being explored. Section 5. Navigating the challenges, side effects and limitations of monoclonal antibodies. While monoclonal antibodies are amazing, they can have side effects. Infusion-related reactions are relatively common occurrences for patients during or shortly after receiving the monoclonal antibody infusion. Patients might experience symptoms like fever or chills. A skin rash or itching can also develop. These reactions are usually due to cytokine release. Doctors are well prepared to manage these symptoms effectively. Because monoclonal antibodies modulate or suppress the immune system, this action can inadvertently increase the risk of infections. Some specific monoclonal antibodies can affect certain organs. For instance, cardiotoxicity or heart problems is a known risk associated with a few monoclonal antibodies used in cancer. Immunogenicity presents another significant challenge with monoclonal antibody therapy. Cost is undeniably a major hurdle for monoclonal antibody treatments. Section 6. Key insights and cracking the code monoclonal antibody nomenclature made easy. To summarize, monoclonal antibodies are truly powerful medicines. They represent a class of highly targeted medical tools. Their remarkable specificity is one of their key strengths. They are designed to attack disease with pinpoint accuracy. This precision helps to minimize collateral damage to healthy tissues. It is helpful to understand monoclonal antibody naming conventions. The suffix at the end of a monoclonal antibody's generic name tells you about its origin or source. For example, OMAB indicates the antibody is entirely murine, or mouse. The suffix, chimab, means it is a chimeric antibody, part mouse and part human. Then, zumab signifies a humanized antibody, mostly human. Finally, umab means it is a fully human antibody. The middle part of the name often hints at the target. Section 7. The adventure ahead. Your future with monoclonal antibodies. The story of monoclonal antibodies is far from over. New discoveries and advancements are happening quite often. As dedicated medical students, I encourage you to learn more. This rapidly evolving field needs bright and curious minds. Your future contribution to this area can be vital. Current research is constantly pushing the existing boundaries. Bispecific monoclonal antibodies are particularly exciting right now. Antibody-drug conjugates, or ADCs, are also very promising. These are just two examples of ongoing innovation. The future for monoclonal antibody therapy looks incredibly promising. Study hard, stay curious, and make a difference.